So it is a pleasure to be able to be here today and share with you some aspects of EV that I didn't really consider to be very important at the start of my career, and that is EV sample collection. I don't think I really fully appreciated the importance of this. This is an education talk, so I've included my disclosures here, but only one is paying and none of them are related to what I'll be presenting today. So I think this is pretty intuitive, and it goes probably without saying, but sample collection is fundamental to anything we're going to do with extracellular vesicles in terms of downstream analysis, in terms of isolation. But recognizing that and then appreciating what that means from a practical standpoint are two very different things. And the question of how to collect samples has really been an issue with the field probably since the inception. And if it were simply a matter of finding one universal sample collection procedure, then we probably would have already reached this by now. But in fact, there really is no single universally accepted way of isolating extracellular vesicles. And really, we're going to be talking about a number of considerations for your own individualized approach for isolating extracellular vesicles. And the reason why there's no single approach has to do with the breadth of what we're doing with extracellular vesicles, the breadth of downstream applications, the breadth of what we want to study. And for one thing, we may be studying various classes of extracellular vesicles. We may be isolating from different biofluids, from blood, from urine, from cerebrospinal fluid, and all of these have different considerations when it comes to sample collection. We may have different downstream analyses, and again, these can impact sample collection. And then, of course, some individuals may want to store the samples, whereas some others may want to analyze their samples immediately. And again, this impacts on your decisions when it comes to sample collection. So, while isolation protocols do vary, I think one of the things we do need to be working towards is a unified approach and a comprehensive approach to documenting aspects of sample collection. This is really fundamental for repeatability and for being able to compare between labs. And there is a number of guidances out there on this. Josh did a nice job of covering some of these, and I think other speakers will as well. In terms of guidance on sample collection, one of the best items out there right now is the ISEV2023 guidelines. If you go to the ISEV website, they have this really nice link to the ISEV2023 checklist. And within that checklist is a number of items that should be considered for sample collection. There's also, other elements of EV analysis, but specifically with respect to sample collection, there's a number of items that would be considered essential for considerations, as well as, context dependent. And it does a really good job of outlining these. So I would encourage anybody new to the field to check out this checklist. So let's focus on some of these considerations that have been talked about. One thing that is essential for all studies is the source consideration at the time of collection. We need to be documenting as much as possible factors that can impact EV biogenesis, factors that can impact EV properties, so that we can link these down the line. And this is something I didn't really appreciate a lot at the start of my career. I can't tell you how many times I would think I documented everything, and then somebody would come back and say, was your patient taking this drug? Or, were your animals, what light cycle were they on? And I think it really is a matter of documenting absolutely as much as possible. When you're talking about human donors, document demographics, clinical characteristics, as many are as available. Animals, you want to be documenting animal biochemistry, housing conditions, etc. Cell culture, source of the cells, whether you've tested for mycoplasma, passage and population doubling time. All of these things are fundamental, and I think generally the rule here is as much detail as possible. Another major consideration, regardless of the source of EVs that you're collecting, is the potential for artificial extracellular vesicle sources. A major consideration here is bodily fluids and even media can have detached or free cells, and these can artificially generate EVs if not handled properly. Generally you want to remove cells from the fluid as quickly as possible to avoid this. In urine, a lot of us consider the fact that this may already be happening with detached cells that sit in the bladder even before urine collection, and how to address this is still sort of an issue of ongoing discussion. With respect to plasma, platelet activation is a major consideration here during and after collection, because platelet activation can artificially induce platelet to EVs. With respect to isolating tissue extracellular vesicles, there are a number of protocols out there for isolation of extracellular vesicles, but we need to consider the fact that simply the tissue dissociation process in the first place can lead to artificial EV generation. And then I think many in the room are familiar with the idea that FBS as a media additive can contain artificial extracellular vesicles. What is probably a little less well appreciated is that other media additives can also contain extracellular vesicles, and we've seen this with certain endothelial cell growth supplements and other items. And so making sure that you're doing the proper analysis to make sure that this is not sort of artificially impacting your results at the time of collection is really important. The timing of collection is also an important consideration. Probably not as much of a concern for cell culture studies. Really, you're probably thinking about confluence in terms of timing a collection there. But when you get into biofluids, this is really important. There are a number of circadian rhythms that can impact EV formation. Simply, a morning void urine tends to have more extracellular vesicles than other urines throughout the day. The true extent to which this is impacting EVs, I don't think we really know this at this point. So, the general advice here is to standardize where possible and as much as possible and document everything, because that's really how we can sort of get to the root of this. Temperature is another important variable. This is a major consideration for storage, which goes beyond sort of the purpose of my talk here, but it is an important consideration at the time of collection as well. For certain downstream applications, avoiding microbial contamination becomes important, and so chilling your samples can have a major impact on limiting microbial growth, especially if there's a lag between collection and processing of your sample. This is not a universal thing, though. You don't have to chill your samples. If you're going to be processing them immediately, if you're going to be doing an analysis on unprocessed sample, it may not be important to chill your sample, so it really is an assay-dependent consideration. Preservatives is also something that may be employed for specific applications. I think probably the most common usage is in downstream protein analysis with protease inhibitors, but this is really assay-dependent. Some people suggest antimicrobials such as sodium azide as an approach to reducing bacterial contamination, but I don't think there's really clear guidance on this right now, and there has been some discussion about potentially using cryoprotectants along the lines of what you would do for cryopreservation of cells, but again, the data in this area is really limited, and so in general, the effect of preservatives on EVs is understudied. I think it's something we need to monitor and keep in consideration. But at the moment, I would say, being careful to consider why you're using a preservative with respect to your EV isolation and what impact this might have on downstream analysis. And finally, with respect to sort of universal considerations, is simply the collection vessel, which can be a consideration. On a very simple level, is your collection vessel compatible with your storage or processing steps? A good example of this is some Eppendorf tubes are not compatible with 100,000 G centrifugation. So if that's going to be a processing step, then you need to consider the tubes that you're using for sample collection carefully. The other consideration is that certain plastics may bind EVs, and again, this can lead to loss, and so just being careful about the sample collection containers that you're using and paying attention to this is quite important. I'm going to talk a little bit about some source-specific considerations. I think this will be at times discussed throughout the meeting, but blood has a number of important source-specific considerations. Now, because blood cells, particularly platelets, can become activated and release EVs during sample collection and handling, protocols should be used that prevent this from happening. So a large gauge needle is typically recommended to avoid cell or platelet activation. Removal of platelets, red blood cells, or white blood cells as quickly as possible is typically necessary. And generally, plasma is preferred here versus serum due to the fact that EV formation, artificial EV formation, happens during the clot formation process. If you are preparing plasma, then the choice of anticoagulant becomes important. Certain anticoagulants can interfere with downstream assays. The classic example is heparin interfering with the polymerase chain reaction. And then the other thing is that depending on the anticoagulant you're using, the anticoagulants may actually dilute samples, and if you're dealing with absolute concentrations, then this needs to be kept in mind as well. Blood also is known to have high concentrations of non-EV particles like lipoproteins that may impact on EV size, again at the time of collection and particularly at the time of processing. This is an important consideration, as well as albumin and other plasma proteins which could impact on downstream analysis like proteomics. With respect to urine, the major consideration here is that urine concentration is highly variable. It's dependent on fluid intake, diet, time of day, and this means that with urine collection, you need to consider approaches to normalization. And so that may mean a timed collection, like a 24-hour urine collection to standardize from person to person. Could also mean adjustments to urinary creatinine levels. And there's some good guidance on this in the urine position statement. TAM-Horsfall protein is a major protein in urine. It's also an issue for downstream analysis because it can impact proteomic analysis, a lot like albumin can in plasma. It's also been shown potentially to bind EVs and have an impact on EV concentrations as well. And then urine is highly at risk of bacterial contamination, especially if there's a lag between collection and processing. So just to briefly summarize, sample collection is absolutely essential to EV research. I think this kind of is intuitive, right? If you have garbage coming in, you're going to have garbage coming out. And so ensuring that at the very genesis of our EV analysis, we are doing the best to sort of control variables is really important. Unfortunately, there's no universal sample collection protocol. If there was, this lecture would have been a lot shorter. So we need to tailor our protocols to the individual conditions and ensure that documentation is extensive. And I pulled this sentence from the upcoming MySEP draft, so don't take it as gospel, but I thought it really does speak to neat considerations we need to be doing at collection. So our fundamental recommendation here, as for studies on EVs from all sources, is to perform well-designed studies and give due consideration and report influences that the methodology to collect, store, and process EVs may have on EVs that are subsequently collected. Now, before I leave you, I thought I would quickly mention a number of resources. Josh has covered this as well, so I won't go into too much depth, but EV Track is really this reporting tool. Is a wonderful resource to help you thinking about the types of variables that you might be needing to consider at collection. It has detailed information from studies on a wide range of bodily fluids, some fluids that I didn't even know humans could make. The other option is to seek out biofluid-specific references. So the Urine Task Force has been very active in this area. There's a couple of documents there. The Blood Task Force has also been very active and has a lot of good information there. And stay tuned because, as has been mentioned already, ISEV has several groups working to improve this across many different disciplines. Thank you.